Experimental pharmacology is a lab-based course that's a part of the basic science curriculum of any medical uh, course. Okay? So during the course of this lecture, we are going to understand experimental pharmacology um, based on two ideas. So we will try to understand experimental pharmacology based on these two ideas. And what are these two ideas? Um, the first idea is that there is order in the universe. Okay? And the second idea is the idea of evolution. Okay? So a discussion of these two ideas would lead us to the understanding of experimental pharmacology. Okay? So let me grab a graphic. Okay, so the first idea is that there is order in the universe. So what is meant by that? It is meant, what is meant is that there is increasing complexity in the world, and that begins from the very basic of particles. So this graphic, if we look at it, we understand that elementary particles, they eventually aggregate to form atoms. Atoms become molecules. Molecules aggregate functionally to form macromolecules, and macromolecules eventually become the structural and functional unit of life. That's the cell. And cells aggregate to form tissues, and tissues form organs. Organs form the organ systems of the body. In the human body, there are 11 organ systems. Eventually, the organ systems give rise to the organism. Okay? So, an aggregation of organisms form the population, and populations form the community. Communities comprise the ecosystem, and different ecosystems make up the biome, and eventually, many biomes make up the biosphere. So, this hierarchy, this increasing complexity, lends to the paradigm of disease. So, disease is what we are all very familiar with. So, the organism is not able to perform um, to the optimal ability. Okay? So, we say that the organism has got a problem, and that problem could be due to a problem in the organ system, and that in turn could be a problem in the organ, and that could be a problem in the tissue, and that could be a problem in the cell. And so there can be a problem. The problem could be associated with the biochemistry of macromolecules, or um, it could be um, at the level of the molecules. There could be a problem with DNA. So this is the level at which we are here today. Um, so medical intervention for a disease in an organism is currently targeted at these levels. Okay? You've got treatment at the organism level, the organ system level, the organ level, tissues, cell macromolecules, or the molecules. We are not yet at the level where we can intervene at the level of atoms. Okay? But um, um, there is something called holistic medicine that looks at the organism in the context of population and community, and so there are interventions at that level. Okay? But as far as um, using drugs for diseases is concerned, those drugs are targeted at these levels. Okay? So these are the levels at which we can intervene for a disease. So the intervention could be at all these levels. Okay? Which means if we want to study a disease, we could study at the level of molecules, macromolecules, cell, tissue, organs, organ systems, or the organism. So the organism has got a problem, and you can study that problem at the molecular level or at the macromolecular level. Okay? Um, so that level, if we think about it, um, is a cell-free system. Okay? So cell-free system. So we can study um, the disease at the molecular level or at the macromolecular level independent of the cell. Okay? So you can study the problem um, looking at the DNA or macromolecules like proteins. So you could study at the level of the DNA, or you could study at the macromolecules, which are basically proteins, carbohydrates, lipids, and this being nucleic acids. Okay, so at either of this level, you can have experimental systems which are basically cell-free, and you can have assays to study the disease. Okay? Or you could isolate um, cells from the organism and then study at the cellular level. You could have in vitro studies. Okay? Or you could study it at the tissue level where you have ex vivo studies. So what is ex vivo? You take tissue from um, the animal and you put it into a synthetic physiological solution, and then you study it outside the body. Okay? So that's ex vivo. So you can have um, tissue studies as well as organ studies, ex vivo studies. You could study the whole organ system. Okay? You can study this in situ, okay? Or an in vivo system. So you can study the organ systems and the organism in vivo. Okay? Okay, so if you want to study a disease and you want to study the interventions for the disease, you can have studies like this. Okay? So we are talking about experimental pharmacology. Okay, so the ultimate goal of experimental pharmacology is to is for the person who is studying experimental pharmacology to understand and gain skills which are needed for drug research. So, drug research um, results in the development of a new therapeutic entity. Okay? So, this drug research we could classify um, for the sake of understanding into two phases. Drug research happens in two phases. You've got preclinical research and clinical research. So preclinical is you go from inventing a drug or discovering a drug, um, understanding its mechanism of action, um, and the basic pharmacology, meaning you understand the pharmacokinetics and the pharmacodynamics, and you learn about the dose. Okay, so you understand everything that you need to put it into a human. So that part of the investigation comes under preclinical. Um, it's divided as preclinical studies. Okay, so you do that. Then your drug is ready for the first in human studies. So, clinical research is the research that is involved subsequent to preclinical studies, and when you introduce the drug to a human being. So, first in human studies. Yeah, so whenever human beings get involved, we call it that part of the research as clinical research. Okay? And there is a specialized branch of pharmacology which deals with this aspect, and that's called clinical pharmacology. Okay. Um, so eventually, you prove that the drug is safe and effective. Okay, so that's the goal, to prove that to gather evidence that the drug is safe and effective. And so this after the first in human trials, which establishes the safety, you look at effectiveness of the drug, and so therefore the um, clinical studies. So clinical studies they can be divided into phase 0, phase 1, phase 2, phase 3, phase 4, and phase 5. Okay. So together these studies are called clinical trials. Okay. Um, so by learning experimental pharmacology, um, one gains sufficient knowledge as well as skills that would enable one to um, be productive in the area of drug research. So that's the whole goal of experimental pharmacology, to make one skilled as well as knowledgeable in drug research. Okay. That in turn opens up a lot of career opportunities in the context of drug discovery and development um, areas. Okay. One could also get specialized in niche areas um, within this framework, for example, one could end up as opting for clinical pharmacology as a career. So those are the areas that's covered under. Those are the studies that are covered under um, experimental pharmacology. Okay. So one learns all the techniques and procedures um, in the area of drug research when one pursues this subject. Okay. So the second idea is evolution. Okay. So let me grab one of the graphics. Okay. So let's look at the second idea. Okay. Um, so the second idea was evolution. Right? So this graphic illustrates the theory of evolution. Okay. Um, it is telling you that life originated, uh, this was the origin of life, and life originated approximately 3.6 to 3.7 billion years ago. Okay. Um, so there are gaps in our knowledge of the progression of life. There is a dearth of fossil evidence. Okay. So as we move up, it's geological time. Okay. So this maybe somewhere here is the origin of the Earth, about 4.6 billion years ago. Okay. So first life, uh, the origin was about 3.6 to 3.7 billion years ago, and there is a part of geological time where we don't have fossil evidence, and then we have fossil evidence, and we hypothesize that there was an organism there. We call it Luca. Luca meaning last universal common ancestor. Okay. Eventually, Luca evolved to form the present day uh, bacteria, and later on in geological time, it evolved to form the archaea, which are primitive bacteria. Eventually, they uh, evolved to form the eukaryotes. Okay. So the first organisms, if that's the geological time, the first organisms that evolved on Earth were bacteria. Later on came the archaea and eukarya. Okay, so um, eukarya meaning you've got a cell and you've got a nucleus. Okay, so when the genetic material is enclosed within a membrane, such organisms we call them eukaryotes. Prokaryotes, bacteria, they they don't have a membrane enclosed nucleus, they rather have a nucleoid. Okay? Right, so this is the theory of evolution, okay, which means that life, the different forms of life, uh, have got a phylogenetic relationship, meaning they are related because of evolution. Okay? So this and eukarya, there we have the animals. So let's blow up this area and look at specifically the relationship between primates and mice. Okay. So yeah, so let's say this is evolutionary time, and somewhere here you've got the mouse, you've got the old world monkeys, You got the New World monkeys, and you've got chimpanzees, and then you've got humans, let's say Homo sapiens, okay, our species. Okay. Um, so maybe somewhere here we had the primitive primate. Okay, so we had a primitive primate which gave rise to our line and chimpanzees. Okay. So because this is evolutionary time or geological time, we understand that we are closely related to chimpanzees. Okay, and um, we have got a relationship with mouse, but mouse evolved earlier than us. Okay? So uh, Homo sapiens have evolved last of all according to this tree of life that we are looking at. Okay? Um, so the point is that um, when we consider evolution, that life is related. Okay, so um, let's look at what we mean by this. Okay, so let's say there is Luca, last universal common ancestor. Luca was alive. So what do we mean by alive? Luca could reproduce. Okay, it could. Um, it had biochemical reactions, and it was alive. Okay, and on the other hand, we have humans. We are also alive. Okay, so to be alive, let's look at. We need to produce ATP. Okay, ATP is the energy currency. So we need to generate ATP, and that was true of Luca as well as humans. Okay, so humans and Luca, what could be the similarity? Both are alive. Okay, so one of the essentials of being alive is to produce ATP. So we hypothesized that Luca could produce, because Luca was alive, Luca could produce ATP. Humans are alive today, so humans produce ATP, meaning the genes which are essential for producing ATP, the pathway that gives rise to ATP. So Luca had genes which coded for the biochemical pathway that produces ATP, and humans have got the same or similar genes. Okay. Okay. Um, okay. So after Luca, we had bacteria. Okay. Um, and so we want to study the energy generation pathway. Okay. So that's our goal. We want to study the energy generation. Okay. So we could use all the organisms which are alive to study energy generation, meaning we could study the energy generation process in the bacteria or the mice or higher organisms um, because we are all related phylogenetically. Okay? So each of these organisms that we could use to potentially study a particular physiological or a pathological problem, we could use multiple organisms. Okay? So instead of studying humans, we could study all. We could study other organisms based on what exactly we are studying. Okay, so all those organisms which are there in the evolutionary pathway could be model organisms. could be potential model organisms. Okay. Now, that depends on the kind of study that you're doing. For example, as we discussed, energy generation, you could use bacteria. And if you want to study, say, contraceptives, so you're looking at, say, it's female contraceptive, female hormonal contraceptive. So the best organism that you could use um, to study uh, the action of hormonal contraceptives would be an organism that has got similar reproductive system as humans. So if we look at this particular tree of life, we could understand that the organism that is closest to us would be the chimpanzee, so that a chimpanzee or a monkey could be one of the best models to study contraceptives. Okay. So we began by saying that experimental pharmacology would enable us to study or understand or make a career in drug research. And in the second part, we looked at evolution and we understood that for different types of studies that we intend to do, we could use model organisms. Okay? And we understood that experimental pharmacology, there would be different types of experiments that could be done to understand drug discovery, to study interventions, etc. Okay? So the kind of studies that one would learn in experimental pharmacology um, as we said, depending upon the kind of model system, we could have um, in vitro studies, we could have ex vivo studies, we could have in vivo studies, um, so where we use whole organisms as an experimental model. So these studies, in vitro studies and ex vivo studies and in vivo studies, they could again be classified as qualitative or quantitative experiments. Okay. So qualitative experiments is looking at whether a drug or an intervention has got an effect, and you answer the question, is it effective? And you get an answer, and those experiments that give you those kind of answers, they are called qualitative experiments. Okay? Quantitative, on the other hand, quantity, you understand how much, okay? What's the amount of biologically active compound present in a particular um, an extract or a crude drug product. You understand it by doing quantitative experiments. Okay? So one of the most common quantitative experiment that one does is are called bioassays. Okay? So in experimental pharmacology, one would learn bioassays. One would learn all this kind of experimental techniques and experimental models. One would learn about bioassays. One would learn about clinical trials um, and other related techniques that would eventually make one skilled in the area of drug research. So that, in summary, is what experimental pharmacology is all about. So if one pursues a course in experimental pharmacology, one would be at the end of the course would become knowledgeable in the areas that we discussed below. Okay, one would learn about pharmacological assays. Okay, different types of assays. Are they qualitative and quantitative? One would learn different techniques. One would learn preclinical studies and clinical studies. One would learn clinical trials. Okay. In addition, one would learn the basic preclinical drug development techniques, which encompasses areas which is usually dealt under the subject of pharmaceutics. Okay. So therein, um, one would become knowledgeable in drug research, and that is what this course is all about. Okay. In subsequent lectures, we would be taking up these areas that we outlined in this lecture um, in some detail. So, um, hope you have an understanding of what experimental pharmacology is all about the scope, and how it would be um, fitting into your future career plans. Thank you for your attention.